and welcome to Year 1 or AS AQA Biology. I've been teaching for 18 years. My name is Mike and I'm a head of biology, so hopefully I'll be able to help you through this. I'm going to go into quite a bit of detail. I'll tell you when it's synoptic links, because in year 13 or year 2 you have an essay you need to write, so I've tried to start including it even right now at the start. And we're going to follow the sections as they're laid out here. We're going to focus in on the section in blue, and we're going to look at monomers and polymers. We're then going to move on to cells, then organisms exchanging in the environment, and finally genetics and everything associated with that. This is quite heavy biochemistry, so there's going to be a lot of chemistry involved in this, but this is the building blocks and the foundation that all the other biology builds from. So it's well worth having this in good detail, really understanding it. Please pause the video whenever you need to. You can rewind it, you can go back and stuff like that. And hopefully I'll be able to explain as we go along. The first thing I suggest you do is pause it here and nip off to AQA website and download the specification. That's exactly what we're going to follow. That gives you lots of advice of what we're going to cover, and you'll see I've kind of laid out the order exactly the same as it is on the specification. So if you haven't already, pause here, off you go, download that. That's what we're going to follow. If you can print it out, it'd be really handy as well, so please do. But once you're back, we'll get started. So I'm going to start with a GCSE recap. So this is nice and simple. I just want you to think about the following. So you should have covered this in GCSE chemistry. So you've got monomers and polymers, and I just want you to have a think about what's a monomer, what's a polymer, and then I want you to define what they are. So come up with examples of them. I've said in pairs there because I actually use this when I'm teaching, but obviously think about it yourself. Come up with a definition and example. So pause it here, have a think about it, and unpause it when you're ready to move on. Right, so a monomer is a single unit that larger molecules are made from. So there's the definition of it there. So monomers are basically the building blocks. So a good example of a monomer is glucose. Another one's amino acids. I'm trying to keep them biological links because we're trying to avoid the chemistry here, because I'm a biologist, not a chemist. So we'll stick to all the sort of biology sort of links there. You could also have glucose, and that's going to be making cellulose, which is a polymer. So the one you encounter at GCSE will be glucose to starch. That's a really good example of monomer going to a polymer. And a polymer is just many monomers that are joined together. So it's well worth having a rethink about those GCSE definitions and keeping them in mind, because all the GCSE you learnt, the A-level will build on. So that's why it's good to just recap that and have a think about it. So we're going to look at biological molecules, and there are so many. Luckily, AQA focuses on just a few key ones, but it's always good to have an awareness of others, just in case they ask those tricky exam questions where they do things like introduce a new chemical that you've not heard of, so a new molecule you may not have heard of. The idea is the principle is the same, it's just the name has changed. So always remember that when you're looking at exam questions. And I'll be sharing exam questions with you and going through them as this course progresses. So what I've done here is I've given the specification link, and like I said in the last sort of introduction, that it's well worth printing this off and ticking it off as you go along. So as we encounter things, as we cover things, you can tick them off, and that gives you reassurance that you've covered everything. And also it might show you where additional linkage or gaps are in the knowledge that you've got. So you might want to look back through and go, oh, I really don't understand this, even though I've ticked it off, we've covered it. What was that video? Go back to it, maybe refresh yourself. It's always good to link back as you're doing things. So we've already encountered monomers and polymers, and we've defined them. And then we've got to look at different examples of this. So what monosaccharides, amino acid, nucleotides are, they're all examples of monomers. We've got what the condensation reaction is and what hydrolysis is. I'm not going to dwell too much on that. That's the exact learning objective taken from the AQA specification. So before we move into all the detail there, we have to have a bit of a background look at the chemistry and the linkage between molecules. So the three we need to know, and luckily we don't have to know them in great detail, we just literally need to know of them, are the covalent bonds, which are where atoms are sharing electrons, ionic bonds, where the attraction between the negative and the positive occurs. So you've got salt there, sodium chloride, and you've got hydrogen bonds. Now, those hydrogen bonds are really important in biology. They're the ones we actually focus in on the most because they're the ones that hold molecules together, but they are weak bonds, so they allow us to break molecules as well. And that's a really handy thing when you're looking at biological molecules. It's so important to make sure that you understand the fact that bonds can break and bonds can be reformed, and the hydrogen bonds are the ones we focus in on. So there is, within the bonding, there's three types. You've got to learn them, but be aware there are more, such as disulfide bonds and stuff like that. But we'll introduce those as we move through the course. So the first, as I've said, is covalent, where it's sharing electrons, forming a very strong molecule. The key thing there is it's strong, so they're difficult to break. You've then got ionic bonds, which are the positive and negative force of ionic attraction, and they're weaker than covalent, but they're stronger than the hydrogen bonds. Now, the hydrogen bonds are the bonding formed between water molecules. So hydrogen has got a attractive quantity which will move it closer to the oxygen, as you can see in the diagram there. So we've got the covalent bond between the hydrogen and the oxygen and water molecule, but the hydrogen bond is the force of attraction between the hydrogen molecule and the free floating electrons on the oxygen. Now, again, we don't need to know it in this much detail, but it's handy to understand that there's a couple of little floating electrons above the oxygen there, which the hydrogen is attracted to. And as you can see, we're forming a chain now, because the hydrogen is a polar molecule which is attracted to another polar molecule, and that is what gives water its properties. So water will form a bead if you drop it on a desktop, so it'll make a little sphere almost of water because there's attraction between all the molecules. It doesn't flow like a lot of liquids. It will still flow, obviously, because it's water, but it'll actually have some quite strong tension. And when we get onto the water topic, you'll see how that hydrogen bonds are very important there, and also the properties of water, how important they are. So hopefully that's given you a bit of an insight into the bonding. Please feel free to pause whenever and just make some notes or print off the slides, do whatever it is to help you. But the key thing is I've tried to keep it to really short, sharp bullet points with the key bits identified there so you can find them easily. So what we've got to do now is we've got to learn how we make and break polymers. So remember, a polymer is many monomers joined together. So it could be a bunch of glucose molecules joined together, as we've got in the picture there. There's two glucose molecules. We join them together to make maltose. Don't worry about maltose, what it is. It's just a more complex sugar. But the fact is they're joined together. And the making and breaking of polymers is so important in biology. There's two key terms we need to know. We need to know the condensation reaction, which is the joining together of molecules, and it eliminates or gives out a water molecule. So literally, as those two glucose molecules join together there, you'll have a molecule of water leave, because what happens is the oxygen and the two hydrogens are on those two molecules, as we'll see when we look at the structure of glucose later. And as they join together, that water is liberated from the molecule, and the molecule joins together, as you can see in the further picture there. And we've also got hydrolysis. Now, this is a great word. Hydro, water, lysis, breaking. So it's literally the Latin for water breaking. So it's a reaction that uses water to break the molecule. As we gained water when the two glucose molecules join together, if we put water into the maltose in that picture there, the water will force its way into the bond, that little molecule that's holding the two monomers together there, and will split apart the molecules, liberating them back into glucose. So it uses a water molecule to break or split apart the hydrogen. And that is such a fundamental in biology. We really need to understand that well. Please pause until you're ready to continue. So we've got the idea that we've got monomers which are single units and polymers which are repeating monomers joined together. So that's your polymers and your monomers. And we've also got the idea now, we've got the condensation reaction, water comes out and molecules are joined together, or hydrolysis, you put water in and it splits molecules apart. So there's quite a bit going on there, but this is the fundamental starting point where all the other biology builds from. So it's well worth getting a good grasp of this and understanding it well. From here, we have Metabolism. Now, metabolism is used a lot in everyday terminology. They might ask you to define it, so it's well worth knowing what metabolism is. From GCSE, you'll probably know it's the combination of all chemical reactions within the cytoplasm. I've defined it here as the sum total of all chemical reactions within all cells of the organism. It's not just a single cell, it's every single cell carrying out its chemical reactions, of which there are 40 plus thousand going on at any given time. So there is a huge amount of chemical reactions going on. If you think of respiration in the mitochondria, that's going on inside every single cell that has one. So apart from the red blood cells, there's pretty much every cell in your body which is respiring away, which is going to be carrying out chemical reactions. Then you've got digestion, you've got the delivery of neurotransmitters across the synapse. There is so much going on. So it's well worth just having that definition. It could be potentially a one marker. It's very unlikely, but it's worth just bearing in mind you've got the understanding of what metabolism is. So as soon as they mention metabolism, you've got to be thinking it's chemical reactions within the cells of the organism. And we only got one little bit to finish off here, and then we're going to move on to carbohydrates. So the last thing, and it's going back to your chemistry, is the definition of measuring a mole of a substance. So the mole is an SI unit, so it's a scientific unit of measurement. And we don't need to know it in too much detail, but it's just a better way of having a little bit of understanding about it, and just to be enough to cover you if they do ask about it. So they're not going to ask you to calculate the mole and stuff like that. That's definitely chemistry's problem, not ours, but they might use moles quite a bit in terminology. So just an awareness of it is important. So I wouldn't go crazy with this. I just have an awareness of it. So it's basically the number of atoms within a sample of a substance, and it's based around the Avogadro number. So what we've got is, we don't need to know the number, but that's what it is, and just have a awareness of what it's called. But the key thing that we need to take for biology is molar solution. 1 litre of solution contains 1 mole of the solute. So whatever's dissolved in that litre is 1 mole. So a mole is molecular mass expressed as grams. So I wouldn't worry too much about it. Like I said, just an awareness of it. And it's just getting back to the GCSE and that whole thing about what a mole is. They use it an awful lot in tables and experimentation within exam questions. So just having a brief awareness of it is handy, but I wouldn't go to too much problem with it. I wouldn't make notes on it and stuff like that. Just have an awareness of it. So what I'd like you to do now is, I'll try and share this on the site so you can download it. If not, you can make a quick sketch of this or take a screenshot of this and print it out. Quite happy for you to do that. So this is covering what we've done so far. So it's basically about monomers and polymers, and there's a few bits and pieces about that. So what I'd like you to do is pause the video here. I'm going to actually end the video here for a moment, and I'll start up again with the carbohydrates, so glucose and that sort of thing. But I'd really like you to stop the video here and just fill this in with what notes you got. Then you can go back through the video and see if you've got everything correct, and just see if you've got your notes up to date. So it'll be well worth doing as a quick recap and then move on from there. Okay, pause it until you're ready to continue. Right, welcome back. I hope you found that helpful and you managed to update that sheet completely. If not, please feel free to make sure you make some notes on it and backtrack in your own time. So what I want you to do is think about what are the 3 types of biological molecules you know from your GCSE. So you should have a good awareness of the biological molecules in there, and which picture do you think makes up which. Pause the PowerPoint in the video until you're ready to move on, and we'll start in the top right, then move down to the one in the bottom right, and then finally the longer one across the bottom there. So just see if you can name them and match them up. Okay, so the 3 types of biological molecules are proteins, lipids, and carbohydrates. And how we've got them arranged here is hopefully you managed to see a few little clues there, such as saturated, unsaturated. You may have said fats, which are saturated and unsaturated fats, which is fine, but then the lipids. So the top one there is lipids, and there's two types, saturated and unsaturated. We'll look at lipids in more detail, but saturated is full of hydrogen, unsaturated has got a double bond in, or more than one double bond. But we'll see that later. The one in the bottom there is glucose. Hopefully you worked out by counting the carbons. There are 6 carbons in there. If not, you might remember it's a hexose sugar, so it's a 6-sided sugar. That would have helped as well. And the final one is protein. So 1, 2, 3, 4 there is the formation of a protein. That's a complex process, which we can look at later when we look at protein structure. But the clue there was the amino acids in number 1. So you may have thought, amino acids, that's proteins. So that's a good recap of the GCSE and also introducing the key molecules. We've also got to identify some biological polymers and monomers from what they're made up of. So we've got 3 different things here. So again, having a look at that, there's quite a bit of a clue there in the middle ones. I've kind of given it away a little bit already, but see what you can come up with. So pause until you're ready to move on. So the top one there, monosaccharides and polysaccharides, that should have been relatively obvious with the saccharide bit because it's the carbohydrates. Then, like I said, the amino acids kind of give it away because I mentioned it in the last one, but it's well worth just remembering that they're polypeptides as well, and that's your proteins. And the last one, nucleotides, RNA, DNA. Hopefully you came up with the idea that it is nucleic acid. You might have thought, well, it's DNA because that's what it's called. Or you might have thought nucleotides. No, the whole thing is it's nucleic acid. So it's well worth bearing in mind that DNA, RNA are made up of nucleotides, but they're universally called the nucleic acids. And we'll look at DNA in more detail again later. We're going to focus today on the top one there, the carbohydrates. So, like we did with the monomers and polymers, that's the learning outcomes, the specification link there. So again, we can tick this off when we finish this, and we're going to look at monosaccharides and some examples of it. We're going to look at isomers of glucose, and we're going to look at the bonding between them. We probably won't go into too much detail about the glycosidic bond. I'll mention that more when we do the disaccharides, because we're looking more at the carbohydrates in the broad monomer version at the moment. So you've got lots and lots of different types. So the monosaccharides, fructose, glucose, galactose, then you've got maltose, lactose, and sucrose, and you've got the complex polysaccharides, which are starch. You've got fibers, which is cellulose. It's a bit of an Americanism there, the fibers. And then we've got the glycogen, which is sometimes referred to as animal starch. But again, we'll cover those when we go to the polysaccharides. So we've got to know a little bit about carbohydrates. It's quite a big sort of molecule in importance. It's what we run on effectively, and we also do quite a bit with it, like respiration, and plants use it for storage. Without carbohydrates, we would not function. And without plants photosynthesizing, we would not get starch, which means we would starve to death effectively. Directly or indirectly, we are all reliant on photosynthesis and plants making glucose. So carbohydrates are literally made of carbon, oxygen, hydrogen in different quantities. So the common one is glucose, C6H12O6, but there are loads of other ones. There's 3 you can see there, fructose and galactose being 2 others, but there are loads and loads of other ones. Luckily, we only really need to know those 3, and the 3 disaccharides there and 3 polysaccharides. So we've only got 9 molecules to really learn. And we don't need to know them in a huge amount of detail. We just need to know enough to get through exam questions and stuff like that. Obviously, there's loads of others. There's pentose sugars and stuff, which we will touch on, and we'll have a look at a few of them, but we won't go into too much detail. So the carbohydrates themselves make up about 10% of the matter of the cell. You're not going to be asked that in exam. It's just an interesting fact, and I quite like the fact that 10% of your body effectively is made up of carbohydrates. And it's a vital functioning molecule. Its main thing is its energy source. So it provides us with the energy in the form of ATP through respiration. We're going to look at that when we cover ATP molecule later, but the unit of energy that we rely on to function is ATP. And we will go into more detail about that. It can also act as an energy store, so we can save the glucose for later. We can't store things as starch, but plants can. That's why potatoes, bread, these sort of things are often starchy food because they've got loads of starch in them. We then eat them for the glucose. Our body breaks it down using salivary amylase, and we can then use it in the act of respiration. And plants also use it as a structural building tool in the form of cellulose. Cellulose we refer to as fibre because we can't digest it, but they are all vital functions. We need to know those three functions and recall some examples of it. So energy store, starch, etc., etc. They can also form up larger molecules. So you've got the glycolipids, literally a fusion between carbohydrates, the glyco bit, and fats, the lipid bit. You've also got glycoproteins. That's what I like about biology. There's lots of terminology that makes sense once you understand it. Glyco, glucose, or carbohydrate. Protein, protein. It's a mixture of carbohydrates and proteins. Glycolipid, carbohydrates, lipids. So there's logic to it as well. You've just got to learn that logic. You've got to learn the terminology. And the general formula for working out a carbohydrate is as follows. So you've always got carbon, hydrogen, and oxygen. It's just the amounts differ. And if you see there, you've got the carbons, you've got twice the amount of hydrogens, and you've got the same amount of oxygens. So if you think about it, you've got to just put the number into the end there and then work out the formula. So for glucose, regular glucose, it's C6. So we've got 6 next to the C there. It's H12, so 6 times 2 is 12, and it's O6. So that's the general formula for it. So if it was a 5-sided sugar, a pentose sugar, you'd put 5 in, so it'd be C5H10O5. It's actually as simple as that. So just have a play around with some of those if you're not sure how to do it. Literally put the number in next to the C and the O, and then double it for the hydrogens. So hopefully that makes sense. If not, I've got a few examples later which will make it a bit more sense for that. And it gets a little bit more complex here. That's just the general formula for it. So pause until you're ready to continue. Right, moving on from that, we have the different types of carbohydrates. Now, we've already encountered some of these already, and I've mentioned them, but it's good to just have a look at this in more detail. So just because you know monosaccharides and polysaccharides and disaccharides doesn't mean you know the full groupings. So the three main groups are the monosaccharides, which are the simple sugars, and we've already covered the formula for that. So it's CH2O, and then you just multiply it by the numbers there. And N is usually between sort of 5 or 6, but it can be between 3 and 7. So you've got different amounts of carbons within the molecule, and that will determine what they do. But they all have basic properties. They all have the three basic properties that are as follows. We've got soluble in water. If you get glucose and drop it into water, it'll dissolve. They have a sweet taste. Think sugars, all of them are sweet in some way. Some are sweeter than others, like fructose is very sweet, but they're all sweet in some form, and they form crystals. And if you're wondering what the picture is there, that is glucose, and you can see, if you really want to, you can pause the video here and you can count up the molecules and make sure that the formula works, but there should be 6 carbons, 12 hydrogens and 6 oxygens in that molecule. So feel free to have a look if you want. If not, take my word for it. And the number of carbons is the important thing. So it all depends on those carbons. The 3 carbons are the triose sugars, the 5 carbons are the pentose sugars, and the 6 carbons are the hexose sugars. So sometimes we refer to it as a pentose sugar, such as in DNA. The ribose bit of it is a pentose sugar, or hexose is glucose. That's the most common. So the most common two are glucose and fructose, and we're going to look at those in a bit more detail in a bit. But just have an awareness that they're all monosaccharides, they're all soluble in water, they are all sweet tasting, and they form crystals. That's the key take from that. And try and learn that formula so you can work it out, because it's often a 2-marker in an exam. They'll give you work out the formula for this, or they'll give you a molecule and say, what does it contain? Now you could, like I said, count the number of carbons and stuff like that if they've given you a nice picture like that, but they won't give you the picture like that. They'll only show the key bits and then say how many has it got. They'll say something like this is a monosaccharide, and they'll give the carbon ring maybe, and then you've got to work out the rest from there. They won't have the hydrogens, the oxygens coming off of it. They're quite sneaky like that in exams, so you've got to know the formula reasonably well to be able to work it out. So pause until you're ready to continue, and we'll move on in a few moments. Right, so the second type of carbohydrates, this is getting a bit easier, is it's two monosaccharides joined together. And you can see there at the top we've got two glucose joined together to form maltose, we've got galactose and glucose joined together to form lactose, and we've got glucose and fructose joined together to form sucrose. We're going to learn that in a lot more detail later, but at the moment just have an awareness of those three names, maltose, lactose, sucrose. And have a look at sucrose for a moment before we move on. Can you see it's a hexose sugar, the glucose, and a pentose sugar, the fructose. So we've got a six-sided molecule and a five-sided molecule together. Now, fructose is very sweet. It's often in sort of sweets and sugary drinks and stuff because it's got a sweeter flavour. So that's often we use. Table sugar is sucrose. So that's what we often use to sweeten food and stuff like that. But the key thing we want to focus on at the moment is the disaccharides. So disaccharides are literally two saccharides. So it's a double sugars. They are two monosaccharides joined together. That's the take from that at the moment. So nothing too complex. Don't go crazy drawing them and stuff like that. Just make a note of the disaccharides are two monosaccharides joined together. And the name again gives it away. And then moving on from there, we have the polysaccharides. If you look at the top there, we've got starch. That is loads of repeating units all together. That's all glucose molecules. That is alpha glucose, all linked together in that chain there. We've then got cellulose, which is again glucose molecules, but this time, being a bit sneaky, it's beta glucose. We're going to look at that when we look at glucose in a bit more detail, but you can see it alternates. So starch, all the CH2OH on the same side, cellulose, they alternate, and that changes drastically the structure, as when we get onto polysaccharides you'll see. And then glycogen at the bottom there is all alpha glucose again, so it's regular glucose that we can use, and it's joined in a particular way with side branches, which again we're going to learn later. I'm just introducing this, but it's good to just have an awareness of these differences here. These are the three types of polysaccharides we need to know, and basically they are lots of monosaccharides joined together, more than two effectively. So you've got monosaccharide 1, disaccharide 2, polysaccharide, many ones or many twos joined together. So you can see the starch at the top there, you can say is 1, 2, 3, 4, 5, 6 monosaccharides, or it's 3 disaccharides joined together. Either way works. I prefer to stick to mono and then becomes this, because the disaccharides are a standalone thing. They often become either polysaccharides or get broken down into monosaccharides. And the problem with diagrams of this nature, if you just look at one of the starch molecules at the top there, is they look quite flat. Yes, they're drawn in a particular way to try and represent the 3D nature using the thickness of the bonds there, and also having them go up and down, but actually they are very, very complex molecules. They're a complex 3D shape. As you can see, that picture there is just showing it flat, but when we look at a molymod, and if you get a chance, do this in your school if you can, it's absolutely amazing to see it. You can see it's a lot more complex than the simple flat diagram that you can see in regular books and stuff like that. It's quite an art to see it. And the black carbons, you can see, often people make the mistake and think the carbons are in a full ring. So you can see where the red one is between the two black on the right hand side there. Often people think that's a carbon. It's not, it's an oxygen. The carbon is actually sticking up the top, where in fact in the pictures it's sticking up top, but actually it's sort of sticking out from the side of the molecule, because there's a lot of repulsion between the elements there. So they're all going to be pushing away from each other. So the atoms, let's be 100% accurate, are all pushing away from each other there, and that's going to cause the particular shape of the molecule. So if you do get a chance, have a play around with Molymod kits. They're actually great fun. If not, just sort of remember it's a 3D structure. We just simplify it down to that flat sort of structure there. And what I'd like you to do is have a look at the diagrams here and see if you can spot the difference between the two. So I'd like you to just see what way they're different. So pause it until you're ready to continue. Right, hopefully you noticed it's actually a subtle difference. Where the number one is, that's the first carbon. So numbering the carbons, they start on the right-hand side and work their way around. We'll cover that in a bit more detail in a bit. I'll go through that in more detail. But the number one there on the right-hand side, you can see on the left-hand molecule, the H is at the top and the OH is at the bottom. On the right-hand molecule, carbon one, the OH is at the top and the H is at the bottom. That subtle difference means a lot of changes in the molecules we can form from it. This is alpha glucose and beta glucose, and that slight change there is vital for when we look at the structure of starch versus cellulose. But at the moment, just realize that there is that subtle difference, and it looks like, oh, all you have to do is twist the OH back round. Molecules won't do that. That's quite fixed in where it is. So you've got the OH sticking up or the OH sticking down, and that's a good thing to look out for, because they could ask you, what is this molecule? You need to know that, so look for that subtle difference there. There are lots of ways of remembering it. I always remember it, it's a bit weird way of doing it, but it might help you, is if it's H-O, H-O, H-O all on the bottom, like ho, 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 think like Father Christmas, you can just make a line between them, then it's alpha glucose. If you can't make the ho, 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 because it's up above, that's beta glucose. So it breaks the rules, B for breaks. So ho, ho, ho is alpha. I also think Father Christmas is human, or, equivalent of, so it's an animal, alpha, and if it breaks the rules, it's B, beta. Now, you'll probably come up with a better way of doing that. You probably think I'm mad at the moment, but it does help just to have those stupid little memory clues to do it. So if you can say ho, ho, ho with all of them at the bottom, that's alpha. If you can't, it's beta. We've also got the concept of the monomers forming polymers. So those monomers, alpha glucose will join to alpha and will make starch. Beta will join to beta and will ultimately make cellulose. So bear in mind that those molecules have that quality as well. So they will join together. You don't get alpha and beta joined together because of the structural differences there. They can't do it. So the alpha and the beta are actually quite important how they do things, as we'll find out later, but at the moment we've just got to understand that they are isomers. So an isomer is the same molecule, different shape. That's all it is. So they are isomers of each other. So alpha and beta glucose are isomers of each other, and the difference, as I said, there is the OH group on the carbon one is different, and this means it has a lot of different properties. So as I've said before, ensure you can draw this. So make sure you understand it. This is a common question in A level. Draw the molecule, or what is this molecule? Either way, a good awareness of it is important. You might not be asked to draw it, you might be able to identify it, or you might be asked to draw it. So make sure you can do both. I try and get people I teach in the school I'm in to sort of sketch it almost by rote, so they can just do it. So every day I just say, right, draw alpha glucose or draw beta glucose, and it hopefully brings it in. You don't have to do it in full detail every time. The key thing is you put the carbons in the right place and you put the OH group in the right place. And now, talking about the numbering convention, we always start on the right hand most carbon. So if you look at the alpha glucose there, the right hand carbon is number one. We then go clockwise round and just number them. So it's 1, 2, 3, 4, 5, and 6 sticking up the top there. This is really important because we often say where they bind together. So if we bound these two together, I know I said we can't, but let's just imagine you can, we would probably link them by the 1, 4 carbon there, so it'll link together. We're going to look at that in more detail in a bit, but just have that awareness that the numbering, we often say it's on the 1, 4 or the 1, 6. They're the two common ones. So it's 1, 4 or 1, 6 bonding, and that's where the bonds form. It's literally saying it's the 1 carbon and the 4 carbon are joined together, or it's the 1 carbon and the 6 carbon joined together. So just bear in mind that there is that convention there of numbering, and that is more important when we get onto DNA and the replication DNA, how we make more DNA. So it's actually quite important there. And they are given different properties because of this. So if alpha glucose is made into a polymer, you get starch, which is digestible by humans. And if you put beta glucose into a polymer, you end up with cellulose, which we can't digest. So two completely different molecules have different properties just by that simple change of making the OH move from the one side to the other. And they're very complex molecules again, as I said. So there's the alpha glucose, there's a slightly simplified one. Can you see the convention and numbering there? And and it's got the OH group at the bottom, and beta glucose, when you look at them in monomod kits, look almost identical. You can hardly tell the difference between them, but there is that subtle difference between where the OH groups are. They are sticking out in different points, but you wouldn't be able to really identify it from looking at the images there so easily. It's quite difficult to tell, but they are subtly different because of where the OH groups are. But the key thing to take is the diagrams. Make sure you understand the flat diagrams. Okay, and the glucose molecule itself is not have done that much detail. Remember, it's just where the carbons are and often where the OH groups are. That's the things you have to label up. We don't usually worry about the hydrogens, and we often don't worry about the CH2O6. So we usually just put a carbon and then leave it as that. So just simplify it down, make a skeletal diagram of it instead of a full diagram is good. And as I've mentioned before, you've got the hydrolysis and condensation reaction. This is just recapping that. So condensation, water comes out and the molecule is formed. So you take a monosaccharides and join them together to make a disaccharide. In this case, you take two alpha glucose molecules, take the water out in the middle there, and you can see it now bonds together to make maltose. And if we put water in, we break the molecule apart and hydrolysis occurs and we liberate it back. So this is what happens when we eat food. We take in food in the form of starch, we break it down to maltose, we then break the maltose down to glucose, we can absorb into the body. So just have that bear in mind there. And I'm going to introduce another term here. The bond between glucose molecules is called a glycosidic bond. G for glucose is a good way of remembering it. So as soon as you see glycosidic, you should be thinking glucose, but don't say it's just a bond between glucose. Glycosidic bonds are any bond between carbohydrates. So just bear that in mind. And you can see it's on the 1,4 there, as I've labelled up in my bad handwriting. So we would say this is a 1,4 glycosidic bond. That's what I'd say there. Okay, pause this. Have a quick read of that, make sure you understand it, then get ready to move on. Right, so we've got to know and identify our structures. So what's the general formula for hexose sugar? Just have a think about it, write it down on a piece of paper, and then you can double check by going back through the video here, or you can have a look at the pictures I'm going to share in a second. So what's the general formula for hexose sugar? Should be relatively simple. Think glucose, but take some numbers away from it, and that should make it quite easy. Right, so the general formula for it is CH2O in brackets 2. That's the way of doing it. And we've got two different forms. They're the hexose and the pentose sugar. So again, can you see we've got the fructose 5-sided, glucose 6-sided, and I asked you for a hexose sugar, so glucose is the most common. The mistake people make here is when they're asked to write the general formula for it, they actually write C6H12O6 because they think, oh, it's glucose. That's not what it's asking. It's asking for the general formula for any 6-sided sugar, a hexose sugar. And of the monosaccharides, there are lots and lots of different forms. We're going to know all of these. So the glucose, fructose, galactose, the important ones. That's why I've highlighted them there. But deoxyribose and ribose are very important when we get to DNA and RNA. They are very similar in structure, as you can see there. It's just the arrangement of the oxygens and the hydrogens on the base there. But again, causes a lot of difference to the molecule. And you can see deoxy is named logically because it's lost an oxygen from the hydrogen on the bottom there, whereas ribose sugar is not deoxy because it's got an oxygen on there. But one makes DNA, deoxyribose, one makes RNA, ribose. But they are both 5-sided sugars, as we'll see later. So to finish off on, it's been quite a bit, but I've got some exam questions I will share with you and you can give them a go. I'll include the mark schemes there so you can have a go at that later. I'll put it into the course site as we go through. But what I'd like you to do is just do a quick verbal or written one here. So Preparation is the key. What I want you to do is just have a quick look at these exam questions. There's only 3 of them. You might want to sketch the top one or just write down where it says. So use the diagram to explain why lactose is described as a disaccharide. That's first mark. It's only 1 marker. On the diagram, draw a ring round the chemical bond where the hydrolysis it's hydrolyzed by lactase, which is the enzyme that breaks down lactose, and the molecular formula for it is C6H12O6. What's the molecular formula for lactose? So you should be able to work that out from there. That's the most difficult one. It's a 2 marker. The other 2 are 1 marker. So pause until you're ready to go. Okay, so for question 1, it's made up of 2 monosaccharides. Simple as that. So a disaccharide is 2 monosaccharides joined together. You could have said 2 single units joined together, that would be fine, but try and use the correct terminology. That monosaccharide is really helpful there. To draw a ring round where it is hydrolyzed, you've got to think, well, we've got to break a bond because hydrolysis is breaking using water. So the area it'll break it is at the oxygen there. That's what I'm doing. And this one's quite a tough one. The molecular formula for lactose is C6H12O6. So a lot of people just sort of go, well, it must be the same for the other one because it's a 6-sided sugar. It's not quite the same because you've taken water out. So lots of people just go, well, double it because there's 2 of them. That's not it because you've taken water out. So yes, you do double it, but then you've got to remove 2 hydrogens and an oxygen. So it's not C12H24O12, it's actually C12H22 because you've taken 2 hydrogens out because of the water molecule, and it's O11 because you've taken 1 oxygen out due to the water molecule. So that's a sneaky one. So that's why it's a 2 marker, because it's actually going to push you a little bit to think about it. So many people when they do this go, it's C12H24O12. It's a common mistake to make, but think the water's come out because it's been put together by condensation. Condensation, water comes out of the molecule. So therefore it is just removing that 1 molecule of water from it. And the last thing is another quick exam question is identify and describe the 2 isomers of glucose. So again, you've just got to make sure you can identify them, describe them, and draw rough pictures of them. The key thing there is it's that OH group is the other way around. Remember that. And as a last question, what has to happen to the beta molecule for the 1,4-glycosidic bond to be formed? So think about it. What do we have to do to beta molecules if we want to join the molecules together? So have a quick think about that. That's quite a complex one. I'll give you a moment to mull it over and see what you come up with. So pause until you're ready to continue. So to form a beta molecule of it, you're going to have to flip the molecule upside down because the OH groups are opposite each other. So each other molecule is going to alternate. So with the alpha glucose, we'll focus on that first. If you look, the OH groups are both on the underside of the molecule. So at carbon 1, where it's circled, and carbon 1, 2, 3, 4, where it's not circled, opposite it, the OHs are both on the underside. So you can line them up and the OHs can join together. But if we look at the beta glucose, the OHs are diagonally opposite each other. It's on the top on that side. So to get the OHs next to each other, you'd have to turn the second molecule upside down, and then you have to turn the next molecule the right way around and the next molecule upside down. Now, this is jumping ahead a little bit to cellulose, but that's exactly what happens in cellulose. If you remember from the picture way back where I said, look, it alternates, that's what you have to do. So you have to flip every other one 180, and that's really important when we get to it. Right, what I suggest you do now is pause the video and you can get on with the exam questions. The mark schemes will be provided and we will carry on with carbohydrates and their structure in the next session, because this is going to take us up to the hour. So I'd like you to do those questions and we'll have a quick review of them at the start of the next session. Thank you. Bye.